I do want to make you aware of some things coming up. Wild Planet Day here at the Great Valley Museum on September 21st. That's brought to you by Modesto Area Partners in Science. And there will be lots of booths, things to do, lots to learn, lots of cool science activities for kids and people of all ages. So I hope you can join us for that. And Science Night is coming up. That is at the Great Valley Museum. We have those coming up. Is the first one's October 4th? Awesome. So October 4th, 7 to 10 p.m. And that's a fun night in the museum. You can go to a planetarium show. You can see things through telescopes far, far away. So I'd love it if you guys could visit us that night, and the museum's open. And then also coming up let's talk about maps first. We have Sean Lyons coming. He's an ornithologist that has traveled the world studying birds. He will be here on September 20th at 7 p.m. over in Sierra Hall 132. So it'd be great to have you come and join us then. Try to get extra credit from your teachers, maybe mention it. I know everybody likes extra credit. And then on September 25th, next week in this room, we have a talk about nursing and what kinds of fields of nursing are out there. And our speaker, and now I can't read it on here.
Oh, full view.
Yeah, the full view is much better.
Still kind of hard, huh?
I think. Let's see. Allison Piretti, and she is an instructor in the nursing program here at MJC. So come out for that. And that's everything, which is terrific, which brings us to Georget today. I am pleased to introduce Georget Oraha. She is going to speak to us today on Tethered through Turbulence.
A little pun.
This is going to be exciting. I missed this talk. This was a MAPS talk that I heard was fantastic, and I missed it last spring, so I'm excited to hear it today. We are lucky to have Georget. She is one of our newest faculty members in biology. She teaches aquatic bio, kind of bringing that class back online when she came to teach for us. And I had the thing to meet her first in the hiring committee. And so really excited about things that she's been doing in the classroom. She went to CSU Stanislaus, graduating in 2017 with her bachelor's in science in biology. And then she went down to Fullerton. So she left the Valley for a little bit, studied marine biology at Fullerton, got her master's degree, and then guess what? Came back to the Central Valley, which I love. And she's now serving her community teaching here at MJC. I asked her if she has any hobbies, and she mentioned her fiance Yvonne. So they are getting married this next year,
yeah.
And so we're excited about that. So
Yay.
Will you please join me in welcoming
all the
Georget?
little claps. Sorry, thank you so much. Can we hear me in the back? Give me a little reassuring head nods. Lovely. Okay. So I first wanted to start off a little bit about myself. Like Professor McInnes said, I went to CSU Fullerton, and I had some of my students tell me, they're like, that's not you in the poster. They're like, this is false advertising. So I said, let me throw up a picture of me actually doing field work. This is in the San Juan Islands, and I went 2 summers to the San Juan, and a lot of my work and a lot of my data from my master's thesis was from traveling there and working at the marine lab over there. So this is me out during low tide working in the field. And I like to kind of talk about, at the beginning of this talk, what a marine biologist looks like. So a lot of time it's like that. I have a little transect tape taking population data. Other times my posture is not as good, still doing the similar type of work. This is in Southern California. And a lot of people think, oh, like you go to the beach for work. That must be lovely. That must be marvelous. And some days are great, but other days it's rain or shine when it comes to collecting your data for your master's thesis. So sometimes you're out there in full-on layers. I had to wear like bibs, like ski bibs one time because it was just so cold out there by the water. So yes, sometimes clear, nice day, and it looks like this. But other times it also looks like this. So this is also what a marine biologist looks like. So I personally am a rocky intertidal ecologist. This is a subfield within marine biology. A lot of people, when they think of marine biology, they think of open ocean scientists. So people that are scuba certified, people that are swimming with all the big creatures in the ocean. My feet were always on land, so I wanted to stay dry. But I learned very quickly that sometimes when you wave at the ocean, it waves back at you. So a little pun for you there too. So if this is a topic that you're interested in, if you've always liked marine biology, but maybe you're not into the open ocean, there is so many subfields within subfields, so many categories within categories that you can explore in this space. So for me, I was like, I want to do this. I'm passionate about it, but I really want to find something where I am on land at all times. So I use this to also introduce what ecosystem am I focusing on? So a lot of my work, see the bad posture is coming back again. So I worked in the intertidal zone. Again, the subfield that I'm studying is rocky intertidal ecology. So there's a lot of terminology and language that we're going to have to go through as this talk progresses. One being like, what is the intertidal zone? And just a general definition, it's where the land meets the sea. Okay, so I'm doing work out here and across all like where there's algae and all the browns and the greens, this is a whole ecosystem right here of living organisms. Think anemones, think sea stars, think barnacles, all types of seaweed, algae, whatever you want to call it. However, there's different conditions in the intertidal zone that make it a very unique environment. There are periods of low tide and there are periods of high tide. When I'm doing work out in the field, I have to be out there during low tide when everything is exposed. So you can see the water is much farther away from me this time, right? And all the living things are visible to the eye and I can do my work. I'm actually doing sea star searches in this space for a monitoring program here. But when the low tide starts to end, we start to transition into periods of high tide. So the water now, as high tide approaches, the water is going to come up and cover up all the living things that I was studying. So it's very much a clock in and during low tide, clock out when high tide's coming. So thankfully we had good technology. I would always check the tides on a little app and I'd be like, okay, water's coming in about 40 minutes. I need to wrap up my work now. So that's a little bit about the conditions in the intertidal zone. So it's very unique compared to other ecosystems because of these high tide and low tide conditions. So what am I actually looking at with this bad posture here? My study organism are California mussels, but a little misconception about the California mussel is that despite its name, it has a very large geographic range. It can be found in the coast all the way up from Alaska down to Baja. So it completely dominates the coast and it serves a very important role in the intertidal zone. So mussels in general, they structure rocky intertidal communities by modifying the environment and creating these mussel beds. So whenever you see a bunch of mussels like next to each other and clumped together like a little group, we call it a mussel bed. These beds can be single layered, right? So if you were to remove the mussel, you would just see bare rock or other organisms, or they could be multi-layered, where there's mussels just stacked on mussels, stacked on mussels. So just a little bit about how they form those communities. You'll notice that when we look at the sketch on the side, this is from a foundational paper that I would have to reference a lot. And you can see when you zoom in to this community, there's other little living things in the mussel bed. You have little anemones, little barnacles, little sea stars, chitons, limpets, the list can go on and on. So these smaller associated intertidal animals really seek shelter in these mussel beds. And these communities, studies have shown they can include more than about 300 species within a bed. And when we think about the environment and we think about, taking care of our planet, taking care of our Earth, a lot of times the poster child for this is the rainforest, right? We've heard about like save the trees, take care of the bees and all that. So when we think about a rainforest, when you go into that space, you have these massive trees and bigger living things that are associated with that rainforest. But if I were to wipe out that rainforest, I wipe out everything else that lives within that rainforest, right? Same idea with the mussel beds. If I were to wipe out mussel beds, I'm wiping out a large community of organisms that reside in those mussel beds. So same idea, just on a smaller scale. Now, let's think about how are mussels fulfilling this role in the intertidal zone? What allows them to attach to each other and to attach to the surface of the rocky shore? So, okay, next slide. We're going to talk about how they attach. Now, the image that you see here with the mussel, and you're like, what is that sticking out of the mussel? It's actually its muscular foot. So mussels have this foot that they will stick out to create byssal threads. Now, byssal threads are basically the mussel's lifeline to the rocky shore. It allows them to anchor themselves to the surface, and then they can attach to each other, they can attach to the rocks, and it gives them this nice security to that community. So I'm gonna play this little time lapse, and you might see some subtle movements, but basically what's happening with the foot, it's pumping out this liquid polymer protein, and then once it's done pumping out that protein, it's supposed to harden, and once it's hardened, then the foot will retract and go back into the mussel. So I always like to think of it as like a cement truck pouring down cement. The foot is shaped like a trough, and then that polymer protein goes down, hardens, and then it's secured, its anchor line to the rock. Now, mussels will continue to do this one thread at a time. So they can't make multiple threads at the same time. And each byssal thread, so this was a sped up video, it takes about 3 minutes for a single byssal thread to form. And they'll continuously do this until they reach about 50 to 100 threads. And they'll try to maintain that for their lifetime. A little fun fact about their life cycle is that some mussels, one of the oldest mussels that have been recorded, I don't know if the data has changed, but it was like about 40 years old for an individual mussel's age. And you can actually guesstimate really, the age of a mussel by counting the lines on the shell. So if you see like ribbed, when you look at a mussel up close, you'll see like these ribbed lines, similar to like rings on a tree, right? You can kind of guess the age of a tree when you count the rings. Same idea when you count these little lines on the mussel. Roughly, you can kind of get their age, which is pretty cool. The species that I studied, so the California mussel, which despite its name, like I said, is found all over the coast, it is, as far as I know, the only species that can do this process when it's out of water. So you can see I have a mussel on an acrylic tile, and it's doing its thing, pumping out that polymer protein, and it's exposed to air, right? It's not submerged in water. As far as I know, all other species of mussels have to do this while they're submerged in water. So that makes this species really unique too. Another little video I wanted to show you is a little bit about their attachment. So you can kind of see these 4 mussels in the lab that are stuck to each other. So I have a little story that goes with this little time lapse. So I'm going to pause it right there. So there's a lot of trials and errors that we're going to talk about as far as my master's thesis work. I love to talk about all of those obstacles and overcoming them to kind of get your final product. So since my design was an original design that I built with my advisor and my committee, I was like, okay, if I simulate what I want to do in the lab, I need to have each individual mussel on a tile because I'm measuring things around individual mussels, not a bed. So I was like, okay, I'll place this mussel on the tile. I'll give it like 2 days to spit out some threads and stick to the tile. And I do this with these tanks that I have set up, that environment of the rocky intertidal zone. I come back and then these 4 mussels, they were by themselves. Okay, they were not attached to each other. They found their way back to each other. And I was like, this is the first of many So their attachment is really an incredible system. We've already watched that. Let's pause that and moving on. So now we've kind of broken down what the intertidal zone is. We understand how mussels attach to the rocky shore. So let's kind of work through what they kind of go through during different periods of time. Because we understand mussels occupy intertidal zone, and they periodically will experience low surface, they're submerged in water, maybe they're filter feeding right now, and they're putting down some more abyssal threads. But there's also periods of low tide where the water goes out. Okay, so then we have some sun, and then they're exposed to sunlight and air. This is kind of stressful, so they get a little hot. See my use of GIFs? This is the most artistic I can ever be, right? So something interesting about the body temperature of a mussel is that if you think about their experience during low tide, this is a very crucial determining factor of their survival because they're exotherms. So their body temperature will actually change with the environment. During low tide, the body temperature of an individual mussel can differ from water temperature like 15 degrees. And that can be due to a lot of different environmental factors too. So think about solar radiation, think about wind, surface temperature. So with us, we're endotherms, right? If we go outside, our internal body temperature is going to stay the same. It's not going to match what the weather is. If it's 110 outside, our body temperature is not going to hit 110. With the mussels, we're seeing those dramatic shifts in their body temperature. So this is where my area of interest was. I wanted to kind of understand what's going on during this period of low tide and how it's affecting the mussels and their attachment strength. So we've kind of set the scene. This leads us to part 1 that I call hanging by a thread. So I wanted to focus on stress, and that can be caused by environmental conditions such as temperature. And if we think about temperatures outside of an animal's normal range is going to cause that stress. And if a mussel is faced with stresses that go beyond its ability to maintain a secure anchor line to the rock, then what's going to happen? These individuals might be more likely to be ripped off the rocky shore. And then again, we go back to this analogy of a rainforest. So if they're starting to weaken, then they're more easily ripped off. And then the smaller associated organisms, right, are also going to be left with no shelter, no habitat. So my goal was to understand what affects mussel attachment during low tide, because maintaining strong attachment allows the mussels to really fulfill its role in the intertidal community. At the time, no studies had addressed this concept about temperature and exposure on mussel attachment. So I really wanted to see how much damage can mussels really take before they're literally left hanging by a thread. So as this process goes on, little did I know it was going to also leave me hanging by a thread a little bit mentally. So we're going to get into it in some more detail in a bit. So what is our objective? We have to think about our question. Science is a process. We think about the scientific method, think about observations. What questions are we going to ask? So I'm thinking now, how does low tide, temperature, and exposure affect components of mussel attachment strength? But what components, right? We have to pick a few different things. I had a lot to choose from. And this is really important with research, right? Because if you pick 10 different things, then you're gonna end up writing something for a PhD rather than a thesis, right? So I picked 3, not 10. So I said, I'm gonna look at the number of Mussel threads per day. So how much quantity of threads are they going to release and put down if they're under stress? Are they going to conserve energy and just hold on to what they got? Are they gonna, in a desperate attempt, spit out more threads to maintain a better secure line to the surface? So that was the first component. Next, I wanted to look at single Mussel thread strength, so just an individual thread. To do this, I looked at the thickness of the threads. If the mussels are going to be stressed out under these conditions, are the threads going to get thinner and somewhat degrade? This is a picture of a single Mussel thread under a microscope. I've darkened the image for presentation purposes, and there's components to the thread too. Again, you can ask yourself so many questions by just looking at components of a Mussel thread. This right here is a little sticky pad that we call an adhesive plaque. There's tons and tons of papers just studying that plaque and the strength of how that is made and how it contributes to the attachment strength of a mussel. So the possibilities are quite endless. Now, my last component was whole individual attachment strength. I want to see also, okay, if they're under all this stress, how much force is it going to take for the mussel to be removed from the rock? What I really enjoyed about this part was, of course, having this creativity with experimental design. I loved marine biology. Again, I liked being outside. I liked doing the field work, but I loved doing lab work too. So I really wanted to design something that happens in the field and emulate it in the lab. So not 100% of your projects, if you're ever into research, they don't always have to be 100% in the field. About 90% of what I did was in the lab. So now we got to break down some more terms, some more language to understand the scenarios that I put these mussels through in the lab. So we're going to break trend when I'm designing my treatments of comparing a more low risk day to a more high risk day. With exposure, we got to look at the tide prediction. So the same way that you check the weather, you got to check the tides. And you can for the muscles to be exposed. So they're probably still going to be submerged in water under these conditions for the single exposure. So more low risk week, really. So the first day exposed to sunlight and air, 4 days submerged in water and relaxed. With multiple exposure, these low tides and high tides, this low tide is a wonderful low tide. If I see tides like this, I can have like a nice 8-hour day in the field because the water is going to be far away from me for a really long time. So mussels are definitely going to be exposed when the tide is this low. So with this, multiple exposure, you got 5 days in a row exposed to sunlight and air. So they're going to go through that period of low tide where they're not submerged. So a more stressful week for them, right? They're going to get exposed to more intense temperatures, more stressful environmental conditions. So that is exposure. Now let's talk a little bit about temperature. So again, the trend of the low risk versus the high risk day. A lot of my work was done in the lab. So for ambient low tide, this is what we deemed as like normal conditions. I'm running my experiment in the lab, and when I have mussels outside of their tanks and they're exposed to a low tide in the lab, aka just out of the tanks, I'm not manipulating the temperature in any way. So they're just exposed to normal lab room temperature. But with high heat low tide, this is where some creativity comes in. So I have this like makeshift heating grid that I put together, and it's made up of these rechargeable hand warmers. I don't know if you guys have seen these, but you can actually charge them and set the temperature to different settings. They just do like a low, medium, and a high. And then I wrap them in aluminum foil so that it could create this kind of conduction so that the mussels heated all around the body. I didn't want to do a hot plate situation because then I'm just heating them from the bottom. I didn't want to do a heat lamp because then I'm just heating one area. Whereas like if I had this grid, I was going to get kind of it hitting all parts of the body. So we'll zoom in so you can see there's one mussel on a tile surrounded by 4 heaters. And I was like, okay, is this actually heating up? So luckily we have connections at Friday Harbor. Someone was like, I have a thermal image camera that you can borrow. So people just have like these lovely pieces of equipment you can use. And you can see the heat surrounds the mussel so that it is getting some nice distribution. So we have, again, a low risk temperature situation, a high risk temperature situation. Now, I have to set a schedule for everything in my life, including my lab experiments. So I ran these experiments based on a way that it would be like a tide going up and coming down. So like high and low tide. And I said, okay, I need to kind of set the hours a little bit. So what I did was I simulated a 5-hour low tide, but then I conducted a 2-hour heat pulse in the middle of that low tide. So yeah, mussels are going to be exposed from 9 a.m. to 2 p.m., but there's a group of mussels that are getting heated in that grid from 10 to 12. So that way it kind of simulates the natural environment. So now we kind of I broke this up into pieces because the treatments for my thesis, and they are composed of those scenarios of exposure and temperature. So I'm going to walk through those treatments 2 by 2 so you can see what scenarios the mussels were put through. So we see these words again, exposure and temperature terms. So we have a single exposure ambient low tide treatment. We have a single exposure high heat low tide treatment. So you'll have a group, I would have a group of mussels, they're attached, I give them time to attach to the tiles. And how did I fix this problem of not moving around? I just rubber banded them, wrapped them like a little present, and it worked out just fine. But the rubber band can't be too tight, then they'll get stressed. So you gotta have, it's like they are very delicate sea creatures. So for our 2 treatments here, mussels attached to tiles, individual group of them are going, and they're all submerged right now. And on day 1, you're going to start the low tide, so that 9 a.m. start time. Some mussels are going to get heated in the grid, and some mussels will not. The low tide will continue, and then the mussels will go back in the tanks days 2 through 5 to really simulate that tidal conditions that I showed you earlier with the predictions. With My next two treatments, we have multiple exposure ambient low tide, multiple exposure high heat low tide. Similar idea. However, every single day for 5 days, mussels are submerged, right? But then we got to start our experiment. Low tide begins. Some mussels get heated in the grid. Some mussels do not. The low tide will continue, and then the mussels get put back in the tank. And they're going to go through this every day for 5 days in the lab. So what did I find out? And I'm not going to dive too deep into the results. We're just going to go over major findings because we got more to talk about of trials and errors. So we're going to walk through those 3 components. What I learned with the quantity of Mussel threads, it didn't really matter what scenario we put them through. The number of exposures, excuse me, and low tide temperature treatments didn't really affect that number at all. But this still has weight. I always tell my students, like, 0 is a result. 0 has value. No change has value. It is telling you something about your data and your discoveries. So quantity, it didn't matter what situation you put them through out of those 4. What I did find is with thread thickness is that one scenario stuck out the most out of those 4, and that was the multiple exposure high heat, low tides. So that's the most extreme treatment I had out of the 4. So mussels exposed to multiple high heat, low tides showed weakened single Mussel thread strength. So they were thinner by the end of the experiment. Similarly, exposed to that same scenario, the most extreme one, mussels exposed to multiple high heat, low tides had weakened attachment strength. So that made sense that at the end we have these thinner threads. It's not going to take as much force for them to be ripped off tiles. Now let's think about the big picture of this experiment. And to preface, my heat treatment in the lab was implemented as a 2-hour heat pulse in the middle of a 5-hour low tide. This is a very conservative approach, right? But because if we think about in the natural world, mussels frequently experience high temperatures for longer periods of time. However, we see even with that conservative approach, there was some effects on their attachment strength. And if we think about repeated high temperature, low tide events, that can really harm the community of the intertidal zone beyond the impact of a single organism. If we think about in the context of heat waves, right? Some heat waves can last 5 consecutive days straight, and they have actually led to mass mussel mortality events in the past. The last mussel mortality event that I had seen when I was in graduate school was at Bodega Bay. And when the researchers went out there, they said you go out there and it's like the mussels were just straight up cooked at that point because that heat wave had lasted for about 4 to 5 days, to the point where the mussels were completely gaped open. And then that's a free meal for a seagull right there. Because if the shells are tough, a seagull can't get through that. So then they were just emptied out shells at that site, which was concerning. And these events are going to keep happening more and more. So if I implement this conservative approach, it's like it doesn't take much for the mussel's attachment to weaken, considering what happens in the natural world. So the data really supported the conclusion that mussels exposed to these consecutive low tides with high temperatures had weakened attachment. And then if we think about, this will happen more changing climate. So again, it's like you have your nice little community, and then it can get a little more bare because you're removing a foundational species. Now, this is where we're going to talk about all the obstacles. So I call this part no calm, just storm. What you're seeing in this picture right now is a robotic mussel called mussel. It's a biomimetic temperature data logger. It's of equipment. So a lot of times I didn't think this was going to happen a lot at my sites. So I was like, there's not a lot of people that go to these sites. It should be fine. And I was like, okay, let's see. Put out one logger, come back completely already getting chipped off. This is like $130 piece of equipment that now I cannot get the data from. And I'll talk a little bit more about robomussels, but this is just one of many interferences I had in the field. So I was in grad school between 2018 and 2021. What happened then? COVID, right? My place of work was also closed, right? Because I worked at the beach. So I was right at the end of my master's and I wasn't quite done collecting my data. So I was like really trying to push, okay, I have my collecting permit. I should be able to still go out as long as I socially distance and do all those things properly. And I just was like, well, kept being told, well, your school shut down, so you got to go back from Fullerton to the Central Valley. You can finish your thesis from home, finish everything from home. And I was like, well, how do I do that? So I put this up and it's like, you should work from home, marine biologist. I still had data to collect. I had to figure out something to complete my field work. Once the beaches had reopened, everyone had this feeling of like, oh, I get to go outside again, right? Because that was like a big moment for everyone. So I was like, I'm going to take my lab experiment. I'm going to set it up in the field and see the differences of that data. That wasn't working out too much, right? So I have a cage here and there's like a clean cut through it. That's definitely not done by a hermit crab, right? And then I have tiles that I put out completely smashed, and I even would see tools left in the intertidal. So people would just like smash something with a hammer and then like leave the hammer there. And I find the evidence that it was smashed. People were scraping things into the tiles, like putting dates on things so they could document that moment, I guess, of my obstacles. So I find this to be really important to talk about, because in science, when you're reading papers, you're looking at news articles and things like that of new discoveries, new technology, regardless of they don't talk about a lot of the trials that come with it. You are reading a paper, you're reading the perfect version of that work in that paper. So I like to talk about the things that go wrong and then still like coming out of that, so it's nice to share also the things that go wrong. And it didn't stop there. You think I'm done? I'm not. So this is one of my sites. 2 articles in the LA Times came out regarding my sites as far as over collection. So massive human moving sea creatures from San Pedro tide pools put delicate ecosystem at risk. I had never seen that many people at my site before until the beaches had reopened, when it was like that COVID period. So I go out, I'm completely shocked. I'm like, what's going on? But again, it's like, these are not protected sites. These are non-MPAs. So people can go out and they can collect, but the excess was putting this ecosystem in a lot of danger. I mentioned that mussels sometimes form single layer beds or multi-layer beds. So single layer, if I take out one mussel, that's just bare rock. That's exactly how this site was. There was not layers and layers of mussels to where if I remove one, there'd be another underneath. So the second article comes out because it just wasn't stopping. CDFW, so California Fish and Wildlife, was going out there trying to stop, trying to confiscate the collections. 45 people charged with poaching marine life from San Pedro tide pools. This was more, these amount of citations, it was like 45 in one weekend compared to like 2 in the last 2 years, 2 to 5 citations within the last couple years. So that is a huge jump into the interference. Here's some more pictures from those articles. I saw these gallon buckets everywhere. They're traumatizing me now, because I just remember like all of the urchins and mussels people were taking. One person was actually, they counted like how many pounds she took, and it was 175 pounds in one day, which is insane. Because let's think about the legal limit at this site. So at San Pedro, the legal limit for White Point, which is the specific site that was documented, it's 10 pounds per day per person. So you could take a family of 3 out there and get 30 pounds of mussels, and you're not going to get citations about that at all, because it's within the legal limit. And then you can go back the next day and get more, and that would be fine. So 175 pounds was in so much excess, that's actually beyond what I was allowed to collect with a scientific collecting permit for the year, which was insane to think about. And then again, science always starts with an observation. So I just started taking pictures on pictures to show people what I was going through. So I have one person here, one person here. I was like kind of dissecting the picture. This person with the orange bag was using a gardening shovel, which was lovely, and just scraping the mussels off. And there's another person hiding there. I said, you know, I'm gonna walk. There's another person in that corner. I walked all the way to the end of the site. There was people doing the same thing on that bench over there, which was crazy. I couldn't fathom what was happening. I had half my thesis done. I really needed that other half somehow, some way. To give you just some perspective, this is what my site looked like before the pandemic. And this is a great situation. I'm a happy girl here with, look at all those mussels. I can do a lot of work. Then post-pandemic, this is what my site started to look like. Wow, I know. That's usually like the wow moment. But it's sad, right? A lot of bare rock, honestly, more barnacles in these photos than mussels, right? Those yellow organisms. That was starting to get alarming. I said, okay, I need to just take pictures and document it because it'll still be something useful. I was really starting to feel like I was in the trenches. So me and my lab mate would go out and just like document videos, photos, anything. Hello? This is the face of someone struggling behind this mask. It was in the trenches. I was really starting to feel like, I don't know what to do. Some more professional data. I had the privilege of being able to work for a monitoring program called MARINE, Multi-Agency Rocky Intertidal Network. What's really cool about this program is that they would monitor sites, some sites for over like 20 years they have data for, and they would take photo plots. So imagine you go to a specific spot into your site, take a photo, what's going on there, and then you can come back next monitoring session, take another photo, and then you can kind of piece a timeline. So imagine 20 years of taking a photo at the same spot and being able to monitor those changes in the intertidal zone of your site. So this is one picture of a bench. You can see mussels on the side. There's actually some baby mussels on the top, so some recruitment is happening. And the next picture I will show you is after the pandemic. This is the same rock. This is the same bench, completely cleared off, right? So even those little mussels that are in this section here, we don't see a lot of those coming back, right? They didn't really make it through that moment of the over collections. So then I was like, I need a new approach. And my idea was that, okay, I actually have population data. I was measuring sizes of mussels because I wanted to get some baseline data to really kind of understand the site that I'm working at. So I had all this data pre-pandemic through the pandemic. So I was like, okay, I can measure out, was there changes in the size population? Like, is it now going to be more smaller mussels present versus larger mussels after this situation? And that was how the second chapter of my thesis was born. So COVID-19 related impacts on mussel populations in Southern California was part 2. So I'm just going to show you briefly some graphs about the size classes and how that has changed. So these are my Southern California sites. We have white point, point Furman. On the x-axis, we have mussel shell length category, so those size classes. The y-axis is just the quantity in each size class. We see a nice little distribution of those sizes, and this was before the pandemic. I had during the pandemic. Let's see how that shifts. So you can see, we don't see anything above 60 millimeters in length. So a lot of smaller mussels. So we're now seeing some evidence of size selective, collections where people are targeting and having some bias in how they pick out what they want from the beach. I also, just for extra data, you can see that there was a lot of, mussels just being dumped back into the beach. And I was like, okay, this can support also if I measure these dead ones, these are probably the ones that were confiscated. I want to see if these ones are large too, to kind of support that people are picking out these larger mussel shell sizes. So a bunch right here, this entire, you can see all those dead, these are all dead mussels just in this little bank right here. So I started measuring those. Here's another image of that to show you. And there's not a lot of data here, but it gives you some idea and some perspective, right? So look how we get all the way up to 100 millimeters in length for those dead mussel shells, just at white point. So people are picking out these larger sizes. Now, what's a major result of this second chapter that was a complete accident? So this study really served as an example of the importance, one, of baseline data and observational data, which can be used to evaluate unexpected events. In this case, we have collection activities, including size-selective collecting, which can have long-lasting impacts on the mussel populations and the intertidal community as a whole. So if we think about this type of disturbance, this event of size-selective collecting, but there's other disturbances that happen in the intertidal. We have natural disturbances like waves. We have human disturbances like trampling, right? So just foot traffic on to the intertidal. That's very common in Southern California. There's a lot of studies on that too. So if we stack that event, put another disturbance on top, then our intertidal zone is going to look a little sad. So these gaps that start to form, they're going to expand a lot quicker, which is something that it kind of supported, like, taking care of, like thinking about MPA laws and things like that. Now, is this still relevant, really? Because they're like, some people say, oh, this was kind of done during a peak pandemic time. Is this even like something that we should still be studying? But this wasn't something only relevant to mussels when we think about human impact at the intertidal zone. This was a friend of mine that studied the Sylvicia compressa, which is called the common name is the golden rockweed. And she specifically was looking at the impact of trampling. So just walking out in the intertidal zone and how that impacted that algae. And that algae actually also serves as another important habitat for smaller associated organisms. So whether it was me in 2018 or my friend Megan here in 2024, this study still serves some importance even by looking at different organisms to see that. Now I'm going to wrap this up with some silver linings because we want to end on a positive note, right? This has been a journey, right? I'm here in the San Juan. There's actually 3 people in this photo. Not as much human interference at sites like this, right? And what's nice about understanding these events is that event that happened where my sites were, that is one instance, right? That is one event. However, this wasn't happening everywhere all around the world. It just so happened to be where I was doing work, which was a little unfortunate. But mussel populations were still doing well in other areas along the coastline. So I went to the San Juan 2 summers in a row. So I have a little bit of data to show you how big those mussels can get. And this was during the pandemic, right? All the way to 150 millimeters in length. That's as big as my head. I could scrape it out and make a cute little clutch if I wanted to. That's how big they were. So these guys are doing just fine. And so I have the June data at 2 sites. Again, it's not a heavy amount of population data because I was only there for so long, but you kind of get the idea. And then July, you can see, again, all the way up to 150 millimeters in length. So this wasn't happening everywhere, which is really nice. Just a little bit about some technology. Like I mentioned, those robomussels, these were developed by Northeastern University through Brian Helmuth's lab. He's like a big mussel guy. He actually really coined the term about mussels really kind of forming this rainforest. And these sensors act like mussels because they have these, they track temperatures inside the mussel beds. And that can really help us understand how temperature changes affect mussel populations. So by studying that data collected by these little robots, researchers can gain insights into the impacts of climate change at this community level, which can contribute to our understanding of broader climate patterns and potentially really help in developing strategies. I have a little screenshot of how the progress that has been made. There's like a database now because these robo-mussels have been deployed all around the world, and you can search up the data and you can track body temperatures of mussel beds all around where these mussels have been deployed. And what's nice about this, it has like some nice Bluetooth technology. So I kind of show you a little bit of that. You take a phone, you can set them to record body temperatures every 10 minutes, which is really cool. And then when you want to go back and collect it, you can just like tap the phone, collect the data, email it to yourself, and you have a nice spreadsheet of all that body temperature data. I know I'm cutting close on time, but I have just one more, like a few more little things to kind of wrap up with another little trial for you. Because again, of course, it doesn't end there. I wanted to use these for my, field work, and I read about one of the scientists that worked on some of these prototypes. His name was Fernando Lima. He was based in Portugal, and I got to correspond with him. And I'm like, wow, I get to communicate with a scientist I've been reading about in like published work. And I did like a little grant situation, was able to get like a batch of robo-mussels to kind of try out. And then I would have to send back the data to kind of share what I found. I put them out in the field. There's like 12. Okay. He emails me instantly within like this. It was in such a short period of time. I was like, this is a busy man. He's a big scientist. He's like, oh, there was a flaw in our design. And it seems like seawater seeped into the robo-mussel and it got into the battery. So it completely destroyed the battery. And then he immediately sends another email. He goes, this is our new design. We're going to send you a new batch right now. I get a new batch, and I was like, wow, you're welcome. Like, I helped by destroying that first batch, right? So at any level of science, you're going to have these trials and tribulations. There's people involved in these trials, and then it can lead to something even better, which is very cool. Again, silver linings, okay? So this is marine data. This is the monitoring program. This is a lot of data. We're just going to focus pre and post pandemic. This is percent cover of mussels at one of my sites where that collection event was happening. There's a gap in the data because we were not allowed to monitor during COVID. Percent cover, all this means is that if I were to take a frame and take it out to the intertidal, and I point, if I look at every point on that frame, if every point is a mussel, that is 100% cover. If half of those points are mussel, then it's 50% cover, right? So we want to see 100% cover. So we can see, look over here, we got a little uptrend pre-pandemic, post-pandemic. Closing thoughts. I'm going to put up some QR codes for you guys, so feel free to scan these if you'd like. Now, the first thing I want to talk about is eating mussels safely. I am not going to stand here and tell you you are never allowed to collect another mussel ever again. But it was kind of scary to see people collecting mussels during times that were not it wasn't a safe time to collect, right? So there's this thing called red tides, and then biotoxins can get into the water, which is dangerous. And mussels are the most dangerous because they accumulate kids, they got buckets. They're collecting some things, but it's within legal limits. They're wearing the right shoes. They're not barefoot out in the beach, right? So it's just about practicing things safely within limits. You're allowed to collect within reason for your own health, for your own safety. So that's just some information right there. I'll go back to it, but I do have some acknowledgements on the board, and that is the end of my talk. Thank you for sticking around.